Hello everyone, welcome to my channel. In this video I'm going to discuss about the PCR, polymerase chain reaction. Polymerase chain reaction is a technique to make many copy of specific DNA region in vitro. So that means this technique is happening, we will do it in the test tube, okay, not inside the organism. Okay, so the PCR relies on the thermostable DNA polymerase. Okay, tag polymerase and required DNA primers designed specifically for the DNA region of interest. So, in the PCR, the reaction is repeatedly cycled through a series of temperature changes, which allow many copies of the target region to be produced. So, and another thing we should not forget that the PCR has many research and practical application. So, for example, it's used in the DNA cloning, medical diagnostic, and in forensic analysis of DNA. So, the other part was important in biomedical research, okay, which is our interest. So, the first of all amplifies DNA, which is allow for detection of many or detection of small amounts of DNA. First of all, the second one, rapid and efficient. So, it's a save time, very fast, and resources compared to the traditional method. So the important thing is very fast, we can easily get it, and we can use it rapidly as compared to the traditional method. So the other part, which is very important, is useful for variety of applications such as genotyping or gene expression analysis and forensic analysis. The other part of the video, I'm going to talk about the type of PCR, which is very important. We have 4 types of PCR. So first of all, we have standard PCR that amplifies specific DNA region using primers. Okay, used for specific DNA region. Okay, so the use for gene expression and genotyping analysis. Okay, second type is amplification of conventional PCR. Okay, that uses 2 sets of primers to increase specificity, used for detection of low copy number targets. So the third one is used for, and the third type is the quantitative PCR that allow for real-time detection of amplification. So this type used for gene expression and used for quantification of DNA and RNA. So the last part, which is very important, used at the COVID time. So this type is PCR that amplify RNA, okay, using a reverse transcriptase to convert RNA to DNA. So this type is used for gene expression analysis and detection of RNA viruses. So now we have 4 types of PCR. First one is standard PCR that amplifies specific DNA region using primers, okay, used for the genotype expression analysis. Okay, so the second type is amplification of conventional PCR that uses 2 sets of primers to increase the specificity, used to detection a low copy number of targets. So third one is quantitative PCR that's used for gene expression and quantification. So the last part is the PCR that amplify RNA, okay, using reverse transcriptase. So used for gene expression and detection of RNA of viruses. So under 3 important stage, which I'm just talking in this video, I'm just talking about that shortly, okay, summarizing, not explaining in detail. So we have 3 stages of PCR. The first of all, denaturation of DNA and annealing, and last part is extension, copying. Okay, these steps are essential for amplifying DNA and generating multiple copy for downstream applications in biomedical researches. So first of all, denaturation, annealing, and extension, 3 important parts. So last thing, which is I will explain for you in the diagram, which is the technique how we are doing. So first of all, we have a sample. So sample, we are taking the sample from like for example for detecting the virus, the viral detection. So we are taking the sample from the nasopharyngeal swabs. Okay, sorry. Second one, for example, from oropharyngeal swabs. Okay, so because it's a virus, so we have RNA. This RNA, extraction of RNA, first of all, we need extraction of RNA. So RNA, like RNA taken from the COVID-19, for example. So the important thing is that we are doing what we are reverse transcription. So by using a transcriptase enzyme, we are converting RNA to DNA. Okay, so we have RNA, we convert it to DNA. Okay, so the last step is, after that, what we'll do, we will do in the amplification, copying. Okay, by using polymerase enzyme, by using this enzyme, we are amplifying the DNA to multiple copy of DNA. So after that, we are detecting the real viral DNA. So by the RNA, which is we have taken from the viral, okay, for example COVID-19, so we just convert the RNA to DNA by transcriptase enzyme. So after that, by using the polymerase enzyme, convert the DNA to multiple copy of DNA. So I hope this video is helpful for you, all about the PCR. Thank you very much for listening. I hope, don't forget that subscribe my channel and like this video for making you more video. Thank you very much.